PPT is seen?
Yes, ma'am.
So we will be starting immunology. In the last class, we have visited the site where you have gone through a video, two videos I think. What about the innate immunity? What about the acquired immunity? I don't think so. Have you gone through that videos? Please go through this one. They are very unique. And today we will be starting with immunity and health and the subject, why it is named as immunology. Immunity is the resistance power of our body, any foreign pathogen or chemicals, because in case of venom also, it is not a pathogen, snake venom. When snake bites, the venom is a harsh chemical which is interacting with the cell systems, and they can be neurotoxic, they can be plasma membrane, means the cell membrane destroying, they may be DNA replication stopping activity. So any non-self foreign component may be a pathogen or a chemical. When they are interacting with the cell systems of the body tries to give a resistance against that invasion. Now, the study linked with this resistance power creation of the body system is the immunology. Now, host, who is the host? Here, we are the host. The animal models, the human being, or any cell which are being infected or being encountered with the non-self product is the host, and the macromolecule, foreign macromolecule may be antigen, and that antigens, they may be the bacteria, virus, and the protein component of the bacteria, virus, either whole or the protein part or the DNA RNA part, or, and another portions are the parasites, worms, anything which are not regarded as the self component of our body is regarded as the antigen. Now, what is the main function of immunity? Immunity refers to all mechanism used by the body as a protective behavior against the environmental agent that are foreign to the body. So here in the broader way, we are telling that they are the environmental agent which are foreign to the body. Basically, a constant state of war exists between the pathogen and the host all the time. Whenever the pathogen is interacting, the host means the cell, maybe in vitro culture, there are two systems. One is in vitro, another is in vivo. What is in vitro? In vitro is the laboratory setup condition, and cells are isolated from the living models and they are cultured in the laboratory, and it is called the in vitro system. And what about in vivo? In vivo is the whole body system, live living organism system. There the study is analyzed in a in situ manner, means in that condition how the antigens, how the foreign component, non-cell component is interacting with all the cells, all the chemicals, hormones, all are taking part to give resistance against this pathogenic invasion. We have gone to these sections, that is how the immune system actually works, and immune system, the innate and adaptive. Now, what about the innate immune system? The defense mechanism, that is the resistance mechanism, external defense, internal defense, and immune defense. What about this one? The non-specific defense mechanism is the first line defense, where the skin, the mucous membranes means the inner lining of your mouth, of your gut, of lungs, the internal organs having some inner lining of the cells which secrete some secretion which gives them easy to interact with the foreign air, foreign things. Suppose you are just ingesting, you were just chewing the food and trying to ingest the food, swallow the food. If the food passage is not slippery, then it would create a very painful feelings for the host. So all the time the mucous membrane secretes something, the secretion, to facilitate this passage. And the skin is the first line defense, where it is capable of resist the foreign interactions of the pathogens and chemicals, and the secretions of the skin and the mucous membrane. The second line defense is the phagocytic white blood cells, that is the WBCs, antimicrobial proteins, and the inflammatory response. That is the whenever we have some chronic infections, we visit to the physicians and they give us to prescribe us to check the TCDC, the blood count. So total blood count and differential blood count, where the numbers of the white blood cells, red blood cells, if it varies, it is the signature whether the condition is a infective condition of the body or it is a normal one. So these are coming under the purview of the first line and the second line defense, and the non-specific defense mechanism, not very much specific against any specific pathogen. But the specific defense mechanism, the immune system gives that specificity of resistance through third line defense, that is the lymphocytes, the T cells, B cells. There are several types of lymphocytes, means that T cells have the subsets, the B cells, the immature B cells, mature B cells, we will be discussing their varieties. So lymphocytes and the antibodies. In the era of COVID, you all perhaps know about the antibodies. Antibody is the chemical which is produced inside the body against any foreign antigen. So antigen is the cause and antibody is the result. And immune response given by the antigen, the result, the causative result is the production of the antibody, the first line. It is called the humoral immune response. And what about the innate and adaptive immune response? The innate immune response is conferred by all these elements which is an individual is born with. Means in a very short notice, the protective behavior through the pre-existing cells, chemicals, protein, protein, the body gives the resistance against the foreign invasions. It is coming under the purview of the innate immune system. Innate comes from the instant one. An adaptive or acquired immune system is more specialized, that is, if you are interacting with some specific antigen variety. Suppose you have taken mumps, measles, rubella, MMR vaccine, BCG vaccine in your childhood, and now you are interacting with the SARS virus. These vaccines won't work against this SARS virus. So the infectivity specificity, when has to be dealt with in a very specified manner, then the reason is arising to create a specific immunity, and this has been acquired by the participations of the different populations of the cell. Now go through the cell system. This is the hematopoietic stem cell. Hematopoietic, hem means, do you know what is the meaning of heme? The blood generating stem cells, they are developed in bone marrow, and in the bone marrow these stem cells are capable to be differentiating into different variety of the cells, and these differentiation naturally in the body system, they can be acquired by the help of different chemicals. They are called the cytokines, different chemokines and hormones, growth factors. These are the stimulus which help in vivo, means in the body, the hematopoietic stem cell which are capable to develop, to differentiate into different variety of the cell can develop. And in vitro means in laboratory, if you know the cytokine cocktail, the growth factors, So these are the cytokines. If you know previously which cytokine can give rise to which stem cell from forming the specified populations of the cell, then you can choose that milieu of cytokine cocktail or growth factor cocktail, and in laboratory you can develop that one. Now, the hematopoietic stem cells are called the pluripotent cells. Pluri means multiple. They have the potency to differentiate into multiple types of the cell. Now, the common lymphoid progenitor, they are of these variety: lymphoid origin, myeloid origin, erythrocytes which have the erythroid origin, and from this one another varieties are developed. Now the lymphoid progenitors, they develop into bone marrow, but they mature into the lymph nodes. That is why they are called the lymphoids. And some lymphoid progenitors, they develop in bone marrow as well as mature in bone marrow also. These are the B cells and the T cell. T, B for bone marrow, T for thymus. So mainly the lymphoid progenitor cells develop into B cells and the T cells, and B cells as well as the T cells, they can interact with the pathogen instantly and develop into plasma B cells or effector B cells as well as the effector T cells. The another variety of the lymphoid originated cells are NK cells or the natural killer cells. There are two varieties of the cell. One is granulocytes, another is agranulocytes. The granulocytes possess, you can see, there are granules. These dots are granules. Which granules? These are called the zymogen granules. Whenever there are interaction with pathogen, the inflammatory responses initiates. These inflammation cause the release of the histamine chemical. That is why we take anti-allergic or antihistamines to stop our nosy, running nose, tears at the time of infections. So, these are the signature of the infection. If you look properly to the nucleus, can you see? There are nucleus inside the cells. The granulocyte possess polymorphonuclear origin. Means poly means many, morphonuclear. Nucleus morphology is having uneven as well as multiple in number. They may be bilobed, trilobed, this divided sort of bean shaped nucleus like this. But the B cell or the lymphocyte, T cell, lymphocyte, monocytes, they have this even type of nucleus. The inner boundary, it is denoting the nucleus, and the outer boundary is the plasma membrane. The myeloid progenitor, it possesses the neutrophil, eosinophil, basophil as well as the monocytes. These monocytes, they differentiate into different tissues, and the names are dendritic cell, mast cells, macrophages. The erythroid progenitor, they are coming under the department of the RBC or red blood cells. So these cells, whichever we are discussing about, they are colorless as they don't possess heme, but these erythroid progenitor, they give rise to the heme containing cytoplasmic organization, and in conjunction with blood they can form oxidized form. They can carry oxygen from one side to another side, for respiratory purposes, and means respiration occurs in mitochondria inside the cells. So, the erythroid progenitor, they give rise to the RBCs or red blood cells, and the debris of these erythroid progenitic cells, they have the smaller fragmentous component, and this one give rise to the platelets. Platelets are not the fully grown cells, they are created by the fractionations of the erythroid progenitor cells. They don't contain nucleus and they are very small in size in comparison to other cells. The platelet, what is the function of the platelet? Do you know? Anybody can tell?
Ma'am, helps in clotting of the blood.
Yeah, very good. So different cells have different functions. RBC carries blood, WBCs have immunogenetic reaction causing capacity. The monocyte, they also can directly phagocytose, just engulf the foreign pathogen as well as the infected cell and give rise to the immunological signal triggering. These are the percentage or numbers per unit of blood. You just see, most of the percentage are taken in account with leukocytes called the WBC or white blood cells, and RBCs, red blood cells, and platelets. Platelets are not the whole cell type as they don't possess the cellular organelle perfectly, and these are the granulocytes called the neutrophils, eosinophil, basophils. Now we are coming to the analysis part, how we will just judge the immunological reactions. Whenever a microbe it is interacting, the epithelial barrier, epithelium is made up of the special type of cells which is very much resistant and hardy, and these are giving as a barrier like protection. Suppose these are the inside cell picture and it is the outside cell picture. So, when microbes are interacting, the epithelial barrier gives some resistance, and whenever it is just coming inside the cell, there are multiple type of cells we are talking about. The phagocytes are the cell variety which are capable to ingest, to engulf any foreign pathogen. Inside these cells there are lytic vesicles having lytic enzymes like lysozyme, peroxyzymes. They enact over the foreign pathogen and they are successfully cleaving the pathogen and destroying this one. This is the methodology for clearing the microbe. Another methodology is to releasing different molecules or the granules called the zymogen granules, and release of histamine happens by the polymorphonuclear component of the granulocytes. So these histamines can directly kill the pathogen. They are acting like a toxin type of substance, cleaving type of substance, neutralizing type of substance. Now, another populations of the cell is natural killer cell. The natural killer cell is the cell which has natural capability to just directly interact with the microbe and kill the microbe. So, these cell populations as well as one protein cascades called the complement complexes, they are taking part in innate immunity. Innate means instantly, and adaptive immunity, they it is started sometimes later, and whenever it is started, then the cell types of B lymphocyte, T lymphocyte, and other cell types, they are taking part and antibodies are generated in humoral immune response. These antibodies, this you can see there are Y-shaped molecule designed here. It is the antibody. At first, whenever the B cell become mature in conjunction with the microbe, unnecessarily body don't waste any energy by secreting antibody or anything else. Whenever there are antigen interaction, then the brain gives signal, it is a high time that you have to secrete antibody. Whenever the antibody is secreted, developed, at first they are engraved, anchored over the cell surface and act as a receptor. Directly antigen and interact with this one, and the antigen design, the epitope of the antigen means the specific site of the antigen, it should match with the paratope, the specific site of the antibody. So, epitope and paratope, when they match with each other, the receptor ligand type of attachment is held and it grabs the antigen tightly, and in subsequent stages we will be discussing now how the antigen is resolved. When there are ample amount of antibody produced in the from the B cell, then these antibody will be released in the circulatory system, and through the circulation the whole body receives the antibody, and subsequently the antibody is giving rise the protection behavior. Now the T lymphocyte, it has two populations. One is helper T lymphocyte, another is cytotoxic T lymphocyte. The helper T lymphocyte, it helps the B cell to resolve, and the cytotoxic T lymphocyte of their own can kill the lymphocyte pathogen. So they are called the cytotoxic T lymphocyte, and the behavior of immunological response is cell mediated response, when directly the cell is capable to clear the pathogen, and when the antibody mediated clearance of the pathogen happens, it is called the humoral immune response. Understood? There are two types of immune response. One is cell mediated immune response, another is humoral immune response. The cell mediated immune response, it is comprised with the cytotoxic T lymphocyte, and the humoral immune response, it comprised mainly with B cells, and B cells are triggered and stimulated with the helper T lymphocyte, and when the B lymphocyte is triggered properly, activated properly, they secrete the antibody, and the antibody mediated immune response, just remember the antibody mediated immune response is called the humoral immune response, and the cell when directly take charge, no intervention of antibody is needed, it is the cell mediated immune response. If you look at the time frame, what is the time needed to be triggered the cell mediated immune response and or the humoral innate immune response? So within 12 hours the innate immune response can be initiated, but the adaptive immune response needs some time to get initiated. So the primary immune response, primary resistance is given by the innate immune system. Now the questions coming in our mind, how may vaccine protect us from infections? Because you have perhaps have gone through the idea since last 2 years that vaccines are nothing but the whole antigen, whole pathogen or the component of the pathogen. So how pathogen which are avoided, which has to be killed, which has to be bypassed, why we are just interacting with the pathogen as a vaccine candidate, how we to do, how we to help. This is the basic conception of the immunological study, that how vaccine can give you protection. And next question is, what may actually occur in our immune system following vaccination? Now, in course of vaccination, the memory in adaptive immune response is formed. The first infection whenever you have got the first vaccine dose, the response is slow because the time needed to be responded, the cell participation, it it kills some time frame and the response is quite slow. 10-12 days it may need to give you the full protection. And the second infection, in case of natural infection as well as the vaccine booster dose, why booster dose are given? The reason for vaccination is to incubate the whole pathogen or the component of the pathogen, uh, and the optimization and the sensitizations of the pathogen or its characteristic molecule to the cellular system. Means, as before exam you are reading the matter and you are preparing the matter, and inside the hall you interact with the same matter based questions and you can answer readily. Just think, if you are interacting the same question without reading the matter, what will happen? You need some more time to reconcile your preconceived idea about the subject. Then you have to make your own understanding, what may be, what may not be. In between time, the exam time will be over, so you can't write well. The same phenomenon is going inside the body system. When the memory of the first infection, it is kept efficiently by the memory cells. When for the second time, for the third time, the same antigen is interacting with the cells, the body system, then the body will readily be recognizing this one, and the time lapse for initiating the immunological response to give protection, it will be reduced. That is why the booster doses are given. The principles of the vaccination, just go through this slide. The vaccination, first dose of vaccination is given. This is the time lapse which has to be passed to initiate the acquired immune response. The innate immune response instantly is giving some response, trying to resist this one, but whenever the pathogen titer is more, body can't deal with the innate one, then the acquired one will be initiated. The initiating time is there and it is called the lag phase. This phase where the body is in vulnerable condition and no such immunity is developed. Now, after 10 to 12 days, the antibody developed against the specific antigen is done and the antibody peak is reached. In this case, for the second time or vaccination as vaccination booster dose or natural infection, the second time if the same pathogen or its any biomolecule or biomarker, any product, any component of that pathogen is interacting with the cells, what happens? The memory B cells are already stored after the primary infection or the vaccine first dose. They will be readily recognizing this one and the cells are capable to interact very efficiently, very fastly, and the initiation of the immune response is started very firmly also. You just see, the picture is not very much proper. I will be showing another picture. This period is quite shortened than this period. It is needing more than 10 to 12 days, but this phase of attaining the antibody titer after the secondary infection or the secondary booster dose is quite small phase. Within 3-4 days, the titer of the antibody production is achieved their peak, and the peak height is also higher than the primary one. So, this lies the principle of the vaccination, why the vaccine candidate is being introduced and why the secondary, tertiary booster doses are given to just recapitulate the antigenic property. Vaccine protect us from infection by including the adaptive immune response but bypassing the need of a primary infection. Means in case of primary immune response, the lag period is needed, but the booster dose vaccination as a foolproof one, maybe a single dose vaccination where the memory has been stored and created for further recognition. It is successfully protecting in future for the same antigen. Just remember, you are taking BCG vaccine, it won't give you the protection for SARS infection. So next day I will be continuing. Today I have to sum up. So next day I will be continuing how the lymphocytes take part step by step to give the protective behavior to deal with the antigenic interaction. Okay.
Ma'am, have you taken attendance?
No, now I will be taking.
Okay, ma'am.